Hello, my name is Gerald Meininger. I'm the director of the Dalton Cardiovascular Research Center and also a professor in the Department of Medical Pharmacology and Physiology in the School of Medicine.
I'm Mike Hill. I'm Associate Director at the Dalton Cardiovascular Research Center. I'm also a professor in the Department of Medical Pharmacology and Physiology within the School of Medicine at MU.
We've been very interested in mechanical function of small blood vessels and even large blood vessels in the body, specifically how the structure of the blood vessel relates to its function.
We're particularly interested in how vessels within the vascular system respond to their environment and mechanical conditions, such as might be provided by the pressure within the blood vessels and the flow of the blood. We can take a three-dimensional approach to the vessels using confocal microscopy. What we're effectively doing is taking very thin optical slices as we move the microscope down through the specimen or the blood vessel. We can then put these slices back together and make a three-dimensional movie.
So this is an example of a small skeletal muscle arteriole.
And the first thing you see is these red fibers, which are again the elastin fibers on the outside of the blood vessel. Now as we go in a little bit further, you start to see these green objects that are located horizontally across the blood vessel wall. These are the individual smooth muscle cells whose role is to either contract or relax to adjust the diameter of the blood vessel and in that way control the flow that goes through the vessel. As we go a little bit further in through the vessel, you're now seeing another red layer, which is more like a sheet. And this is more elastin, but this is what's called the internal elastic lamina. One thing you can notice in this layer is the holes. These allow connections to go between the smooth muscle cells and the endothelial cells so they can coordinate each other's activity. As we go further into the vessel, we're now into the lumen of the vessel, and you can see longitudinally orientated, that is straight up and down along the vessel, the nuclei of endothelial cells, and these are the cells that line the blood vessel wall and separate the blood from the vessel itself. Now you're seeing a total cross-section of the blood vessel, so we're effectively optically sectioned from the outside halfway through the blood vessel, and the things that you can appreciate here is that there's an internal red layer, or the internal elastic lamina. Then there's that outer red layer of fibers of again of elastin, very different in their form. The inner layer is a sheet, the outer layer is more of these individual fibers. And you can see the smooth muscle cells cut in cross-section in this particular view, and you can see a couple of nuclei of endothelial cells along the vessel wall.
This image shows a three-dimensional reconstruction of a complete stack of images so that you can get a representation of the actual structure of the blood vessel wall. And what distinctly shows up in this 3D rotation is that there is a very distinct outer red layer, a very distinct inner red layer, and on the outside of the rotational stack of images, you can see the nuclei, in this case of the smooth muscle cells, and on the inside you can see the location of the nuclei of the endothelial cells. And one of the things that really impressed us was the fact that this particular vessel type has these two distinct layers, but these layers are interconnected so that they're not actual independent layers, but they are connected as some type of large superstructure.
The exciting part of this work, we think, is actually twofold. Firstly, that it's very relevant to our understanding of how a blood vessel works. We think of them basically as just being tubes that carry blood to the tissues, but they're very dynamic machines, and being able to understand how the smooth muscle cells are able to contract and regulate blood flow in this very complex three-dimensional mechanical environment is very exciting from a basic physiology point of view.
We have high hopes of seeing this research continue as we bring in additional collaborators with other unique expertise. And one of the features of being here at the University of Missouri is the breadth of expertise that's available on a complete university campus.